Good morning, everyone. I'm Greg Daniel. I'm Deputy Director of the Duke Margolis Center for Health Policy. I'd like to welcome all of you to this fascinating meeting so far. We've heard so much already about how FDA is envisioning the use of the system today and its growth into the future, and also how we heard from Rich, a lot of the activities that are happening under Sentinel, both on how FDA is currently using the system for medical product evaluations, but also, you saw on the slide, a laundry list of methods and analytic tool development. So these next two panels are intended to dive into a bit more detail on both of those things. In this very next panel, we'll hear from two folks within FDA on how the Sentinel system is actually informing CEDAR and CBER operations. We'll be joined by Marcia Reichman, Senior Advisor and Scientific Lead for Surveillance Programs at the Center for Drug Evaluation and Research, as well as Azadeh Shoabi, the Sentinel Lead at the Center for Biologics Evaluation and Research. Sorry about that. I'll go ahead and turn it over to Marcia. If you're welcome to present from there or from here. Okay, thanks. Yeah, you can do it that way or the
Good morning. It's a pleasure to be here today, especially as we finish this year of transition and we move from the Mini Sentinel pilot to the Sentinel system. Okay, so what I'm going to do is talk about the transition from Mini Sentinel. So I'm going to talk about basically things that have gone on during the past year. The transition from Mini Sentinel to Sentinel and highlight some aspects of that. You've heard a bit about the internal consolidation of the Sentinel management in the Office of Surveillance and Epidemiology. I'm going to talk in more detail about the ARIA tools and the process for using those tools at FDA, and also talk about the continued dynamic expansion of Sentinel capabilities, because this transition and completing this transition doesn't mean that you know, as you've heard earlier, doesn't mean that things are now finished. They're very much in a state of continued enhancement of the tools and continuing increasing capability. And part of that comes from, as I said, the enhancement of the tools, increase in data sources, and I'll finish up by talking about some activities that are just starting now and some potential activities. So ARIA, the Active Risk Identification and Analysis System, is a subset of Sentinel capabilities. It's those tools which are semi-automated at the current time and which don't require de novo programming. So these tools are similar in style to SAS procs, and I'll show you later some of the types of parameters that go into these tools, and really want to emphasize how close they are to a complete epidemiologic study. So ARIA fulfills the FDAA mandate for active surveillance. And as I mentioned, the capabilities will increase and the tools will change over time. ARIA takes advantage of a number of things that have gone on during the Mini Sentinel pilot. It takes advantage of the previously developed algorithms for outcomes, for indications, for eligibility and exclusion. It takes advantage of what we refer to as scenarios of the specific situations which we've described in previous queries, and how to reuse those and modify them just slightly for new queries. It also takes advantage of the infrastructure, the data itself, the query processes, and I think, as Rich and others have emphasized, above all, the collaborative partnerships between the data partners in Sentinel, FDA and Sentinel, FDA and data partners. And I look forward to us all meeting together in person more frequently as we move on. So how is FDA using these tools? So the first thing we want to do when we decide if ARIA is going to be sufficient for answering a regulatory question is to see if Sentinel provides enough information so that it's feasible to address the question there. So we want to identify the exposure, the outcome, the eligibility and exclusion criteria. We also want to look at the level of exposure within the Sentinel population and the outcome incidence. Is there enough power? What risk level can be detected for this specific query? And is this level of risk useful to address the regulatory issue? And we want to keep in mind that when looking at observational data, or really any type of data, that the populations that go into a data set can be very different, and one can get different results, and it's not that one is right or wrong, it's just that the underlying characteristics of the population are different. So then, once something is feasible within the Sentinel system, we want to see whether it's feasible to complete an assessment of the issue with the ARIA automated tools. So to do this, FDA conducts a meeting of staff that are involved in the regulatory issue. And this encompasses in CEDAR individuals from a number of different offices, from the Office of Surveillance and Epidemiology, the Office of New Drugs, the Office of Biostatistics. These are always represented. Other offices, such as generic drugs or other groups, are there when the issue is pertinent to them. And so these meetings examine all the data sources that are available to FDA and determine which sources are relevant to the particular issue and which are the best data sources for addressing the issue. Often there's a lot of consolidation of information from different data sources, from FAERS data, from case series, from Sentinel. So then the question is ARIA sufficient to provide data to address the issue, and if not, to identify the gap? So with that, I just want to go over a little bit some of the history of the types of tools that we've used. So the early modular programs had a number of limitations, and there's been a continual expansion of these capabilities, with, you may have noticed, some change in names of tools as we moved along. So the complexity of safety scenarios that we can address has really increased in terms of the pre-existing conditions, concomitant treatments, complex outcomes and exposures. When we started, our outcomes were mostly a straight list of codes. Now we have a huge amount of Boolean logic there. We have different things will happen in a different temporal sequence that will compose the outcome, and these are really quite complex. The analytics has also changed. The initial modular programs did not have much adjustment for confounding. They had stratification, but not really adjustment for confounding. We see that now through propensity scores, and we're beginning to look at inverse probability of treatment methods as well. And also there are sequential analyses. So in using the semi-automated tools, so I want to emphasize the increase of ease of specifying the query and the increase in speed that we get to be able to produce answers, but at the same time, we want to be very careful that we don't lose the clinical and epidemiologic input. And it's really been a pleasure to work with Harvard, because this is a concern through all of the Sentinel partners. So what do the modular programs look like now? So really the basis of the system is the CIDA tool, the cohort identification and descriptive analysis. So this defines the cohort, inclusion-exclusion criteria, exposure and outcome definition. And this gives a descriptive output and incidence rates. And we also refer to this as a level 1 assessment. This information is needed to perform the level 2 assessment, which can do propensity score estimation and matching and gives a real estimate of risk. Level 2 also produces the standard table 1 that we all see in papers. Level 3 operates from the results of level 2 and allows us to do multiple looks and sequential testing. So at this point in time, ARIA includes level 1 and level 2, and as we move towards the future, we'll also include level 3 analyses. So I want to talk about this history and about use of the level 2, level 3 queries within the framework of NOAC assessments. So the initial, so dabigatran was approved in October of 2010. And so the initial queries that related to dabigatran and warfarin used the very early modular programs. And we knew there was a problem early after approval, and so we looked when there was just about 3 months of data at 2 major data partners, the 2 largest data partners, and at that point there was a total of only about 1,000 new users of dabigatran, and so we couldn't do anything. We looked when we had data through the end of 2011, and at that point there were about 10,500 new users of dabigatran who had a diagnosis of atrial fibrillation. And so at that point, it was possible to use a modular program, get some reasonable results, and these results were the subject of an FDA drug safety communication. We actually looked again, having an additional year of data and more individuals, and at this point in time, we were really able to look, for instance, at different age groups. So these modular programs had no adjustment for confounding, but they did allow stratification. By the time we got to the data through 2012, we were able to see the increase in gastrointestinal hemorrhage with age that had been observed in RELY and that had been observed in Medicare. But we did need to have that larger sample size to be able to do that. And also, this brings up the issue of different populations. The Sentinel population is very different, certainly from the Medicare population because it includes those under 65, and also from the RELY trial population. Again, it's much younger. And so the results fit in very well when you take this into account. We're also doing protocol-based assessments to look at dabigatran compared to warfarin, and a level 2 assessment. These both involve propensity score matched cohorts, and there are sensitivity and secondary analyses. In terms of apixaban and warfarin, we've begun to look with a level 2 assessment at the outcomes of gastrointestinal hemorrhage, intracranial hemorrhage, and stroke. The uptake is such that we're not really at the level where we can do much at this point in time. Rivaroxaban, compared to warfarin, is the subject of the first level 3 assessment that we're doing. And there are about 15,000 rivaroxaban new users, most of which have been matched using propensity scores. In this match, as opposed to the matches that have been the primary part of the analysis for dabigatran and warfarin, the match is not 1-to-1, but a 1-to variable match. The average follow-up time is 3 1/3 months. 2 looks are planned, and we're beginning to see results from the first of these looks. So the next several slides, which I'm going to go through very quickly, are there to give you an idea of how complex the level 2 assessments are and how close they are to a protocol-based assessment or a complete epi study. And these slides will be posted, so you'll have time to look at these. But there are enrollment criteria, inclusion-exclusion criteria, information on exposures. In particular, more than 70 covariates go into the propensity score estimate in these studies. Also outcome information. And as mentioned earlier, this really leverages previous work in terms of the outcome definitions and in terms of the definitions used for the covariates. I wanted to show this slide. This comes from the first look because this is a diagram that's part of every level 2, level 3 set of results that FDA sees. And this characterizes the 2 cohorts in terms of their propensity scores. And what this, in addition to the percent of the population new users of the drug of interest that are matched, this also allows us to see the differences in propensity scores and allows FDA to make a judgment as to whether there's any substantial part of the population that won't be included in a propensity score matched analysis, and whether we need to really worry about that and worry about not only what's there in the population that's included, but what's being missed. And you can see the figure on the right shows the match. They're basically, the 2 lines are basically, 2 curves are basically on top of each other. This just shows something similar from a study that has been presented previously where the outcome is angioedema and the exposures are to ACE inhibitors and beta blockers. So I want to talk about the value of the level 2 and level 3 modular programs to FDA because this is really a huge leap in terms of what can be done, the speed and cost effectiveness. So these tools provide data that's very similar, not quite the same, but very similar to the main results of complete epi studies. They allow for adjustment for confounding, some secondary and sensitivity analysis, increasing flexibility, and they allow for clinical and epidemiologic input. So this really leverages the infrastructure and all the processes to allow a faster and more cost-efficient result and provide knowledge which is very useful in making regulatory decisions. Janet and others have commented earlier that there's a lot of money that goes into the infrastructure. And so when you do the first study, it's still hugely expensive. When you do the second, you cut it in half. And by the time you do the 10th or the 20th, you've really leveraged that infrastructure and you don't pay for it each time you do a one-off study. So I just want to emphasize those aspects of it. in terms of limitations, all of this work is limited by the nature of claims data, by the level of exposure to the medical product of interest, the incidence of the outcome. No technique and no program can give a good answer if there aren't enough people using the drug or if there isn't a high enough incidence of the outcome. Since I like to show data, I'm just going to use the next couple of slides to present an update on the drug use in pregnancy work that actually I described last year. These are very heavy data slides. You can look at them at your leisure. I'm just going to point out a couple of features. So in this first slide, can you hear me if I walk away and talk loudly? Okay. So in this I don't. Sorry. So if you look at the column to the right, you can see women who have delivered a live birth, who have pre-existing diabetes before the pregnancy. You can see that there are about 15,000 unique pregnancy episodes out of a total of 1,900,000 roughly. You can see, if you look at the maternal age at delivery, that the women with pre-existing diabetes tend to be older, and they tend to have many more preterm births, and certainly less post-term births. If you look at the lines that are highlighted in yellow, you can see that in terms of use of anti-diabetic agents by trimester among women with pre-existing diabetes, you can see metformin. If you look in the three columns to the right, you can see that metformin use goes down quite a bit from the first trimester to the third trimester of pregnancy. Glyburide, if you look at the two columns to the left and compare the use 90 days before pregnancy with any use during pregnancy, you can see a doubling, more or less. And for any injected insulin product, you can see also a large increase between the use in the 90 days before pregnancy and the use during pregnancy. This last slide just shows ADA use in women with gestational diabetes diagnoses who had no ADA use prior to pregnancy. You can see that metformin is not heavily used, whereas insulin, the use goes up and is quite high in the third trimester, and similarly for sulfonylureas. So I know I'm going way over. I apologize. I want to conclude with two quick slides. In these slides, these data sources have been described by Rich earlier. I just want to mention two potential uses of these data sources. These have not been started, but we've been talking about them. Also picking up on something that Steve talked about, we've been talking with CDC about the use of the Hospital Corporation of America data to track complications of flu, look at the seasonality and incidence of these, and so be able to monitor this and to be able to pick up when something unusual is happening and to be able to be alerted to this as early as possible. In terms of Medicare data, we've talked about looking at the characteristics of the 65-plus population in the Sentinel 18 data partners and comparing this with the Medicare population once it's in the Sentinel Common Data Model. We think that these populations may to some extent complement each other, and also there's the issue of those who are over 65 who work full-time and don't have Medicare insurance at all, and kind of where they fall and what their medical characteristics are like. In terms of future activities, these are two activities which have just started. We've done some work over the past year and a half with the Office of Generic Drugs, looking at switching between brand and generic products, but we're really interested in developing a tool that is really designed to look at this, that starts when a new molecular entity is first used and tracks the pattern of use of various generics and brands from that time on. And also we want to be able to look at tools that are used to monitor health status during use of a medical product, where the example is ECGs during donatorone use. This is a list of just some of the people who've been involved in the specific assessments that I've talked about. And I can't emphasize enough how great it is to work with folks from the Sentinel system, from the SOC, from the Operations Center, from the data partners, and what a great experience this is. And productive. That's it. Thank you.
Great. Thank you, Marcia. I'll now turn it over to Azadeh.
Good morning, everyone. It's my pleasure to be here. And I would like to start by saying that what I'm presenting today is the result of the work of many people at the Sentinel Operations Center and also at CBER. So I will acknowledge them at the end, but just to start with that. I am going to talk about the CBER Sentinel program to give an overview of the Sentinel program integration into our product lifecycle in CBER and also provide an update on some of our current projects. CBER has regulatory responsibility for vaccines, blood and blood products, and also cellular tissue and gene therapy products. And CBER's Sentinel program consists of components for these specific product groups. We have PRISM, post-licensure rapid immunization safety monitoring for safety surveillance of vaccines. We have BloodScan, blood safety continuous active surveillance network for blood and blood products. And we use General Sentinel for cellular tissue and gene therapy products. CBER has a very heavy and large portfolio of Sentinel projects. I will not be able to go over all of these, but we have several protocol-based assessments with a medical chart review component. We have assessments that are using our rapid query tools. We are working on several methods development projects as well as some infrastructure buildings, and I will be providing some examples of all of these four categories. This diagram attempts to show a rough timeline for a product lifecycle and also highlight certain key time points in the lifecycle of CBER products. Let's say that a BLA is submitted to the FDA. It goes through the pre-approval process, safety and efficacy assessments are performed, and then the product is approved and released to the market. During the early post-approval period, usually roughly around the first 18 months, there are certain safety activities that are conducted. And then at the 18 months, again, roughly around the 18 months, there are two safety evaluations done by FDA. One of them is post-market safety evaluation, which is mandated by Section 915 of the FDA Amendments Act of 2007. And then the other one is a post-approval safety review to the Pediatric Advisory Committee for products that have a pediatric indication. And then we move on to the later post-approval period. Again, certain safety activities will be conducted. The point that I'm trying to make here is that Sentinel can be used throughout the life cycle of a product at different stages. Some of the safety activities that may occur in the early period after post-approval may stem from potential signals that are generated from the literature, from our passive adverse event reporting systems, such as FAERS and VAERS, and also manufacturers' adverse event reports to the FDA. And in addition to that, some safety signals may have originated from the pre-market studies. So before Sentinel was built, FDA had certain options for following up on safety signals. Some examples were post-market requirements or post-market commitments. Now with Sentinel, we have access to health data of a large population of the United States, which includes data from both private and public insurers. Our data, as you have heard many times up to now, include administrative and claims-based data as well as access to some electronic health records. We have built some rapid query tools that can query our large database to answer some simple questions, as Marcia mentioned, just related to specific outcomes and exposures, or we can delve into more complex questions and look at the association between exposures and outcomes. And so far, you have heard this point again before, that the focus of Sentinel activities have been on signal refinement and following up on safety signals that were generated from some other sources. Now we are working on our signal identification capabilities, which is a tree scan. So a tree scan is a signal identification method and just a data mining tool. It's capable of evaluating thousands of individual diagnostic codes, and in electronic health data, it detects excess risk grouped into hierarchical tree structures of diagnostic codes. And Martin Kulldorff will be talking about this later today, and this is a product of Martin's work and his team. A tree scan is utilized for ascertainment of medically attended early onset diagnosis after vaccination. It is able to detect unspecified adverse events because it's a signal detection program. And it can also detect rare adverse events because the PRISM has a very large sample size. We have used vaccines mainly for development of tree scan, and we are also using tree scan right now for certain signal detection activities with vaccines. And tree scan also helps to determine post-vaccination risk window. So one pilot study that is using tree scan is for Gardasil human papillomavirus 4 vaccine, and this is a pilot project, and after completion of the pilot project, we have plans to use this capability for HPV-9 vaccine. Now, aside from the tree scan HPV-9 vaccine that I mentioned, we are working on another project. It's a current project of Sentinel that was initiated in the early post-approval period due to some reports that we received from the literature and or detected in the literature, and also some adverse event reports from outside the United States. This is a project to conduct prospective surveillance to evaluate excess risk of 4 proposed outcomes after HPV-9 vaccination, and we will be using our sequential analysis method and query tools, available query tools. The 4 proposed outcomes are complex regional pain syndrome, uveitis, acute disseminated encephalomyelitis, and acute pericarditis. Now moving on to the later post-approval period, some potential sources that could generate safety signals for this period could be 18-month safety evaluations, advisory committees on post-approval safety, and also pediatric advisory committees. Now, the point I'm trying to make here is that some of the activities that we are doing in the early post-approval period can be actually feeding information to us for the later post-approval period activities. Again, for the later post-approval period, before Sentinel, FDA had certain options to follow these signals. Now, Sentinel has been augmenting those available options to FDA, and FDA now itself can perform certain activities by using our rapid query tools. You heard about ARIA this morning. We can also perform protocol-based assessments with medical chart review components. One activity which may be initiated either in the early or later post-approval period is safety of vaccines in pregnancy. And the evaluation of safety of vaccines in pregnancy is critical because vaccines can have an impact on pregnancy outcomes, but more importantly because in pre-market studies, pregnant women are either completely excluded or there is a very limited participation of pregnant women. So it's important that perhaps at other stages of the product life cycle, such as the post-approval period, then this evaluation is performed. So in order to be able to do evaluation of vaccine safety in pregnancy, we first needed to build an infrastructure component in Sentinel. And that was the capability to be able to link mothers to their infants. So we performed a feasibility study for this purpose. 4 data partners participated in this study. They were all large national health insurers with claims data. And the study population consisted of women between the ages of 10 and 55 years who were hospitalized for delivery of a live birth between 2004 and 2012. And the infants had to be enrolled in the health plan for at least 1 day. The data elements that we used to link mothers to their infants, at the first pass, it was the health plan subscriber ID, in addition to the matching between the delivery date and the date of birth of the infant. And then for the remaining unlinked mothers, we also added last name and address to the linking data elements. So these are the results. Although 4 data partners participated in this study, we have results for 3 of them. As you can see here, this study included a large population. The number of deliveries are over 330,000 for 1 data partner, 200,000 for another data partner, and then around 30,000 for data partner number 3. And overall, between 82 and 85% of mothers were linked to their infants. So now with this capability, we can delve into evaluating safety of vaccines in pregnancy more easily. We have a test case currently working on a test case. The exposure is inactivated influenza vaccine, and we are working on 2 outcomes, spontaneous abortion and oral cleft. Now, oral cleft was not necessarily an outcome that we had any concerns about, but it was an outcome that could be more easily detected using claims data and diagnostic codes. So that's why we are using oral cleft for this purpose. And the objective of this study is to build a structure to examine pregnancy outcomes after vaccination, and also to examine the positive predictive value of claims codes for spontaneous abortion, gestational age, and oral cleft. The study design is case-time control design. We have 2 sets of cases and 2 sets of controls. Vaccinated women with a pregnancy outcome of spontaneous abortion, or giving a live birth to an infant with oral cleft. And then controls, 2 sets of controls, vaccinated women with pregnancy outcome of live birth or giving birth to an infant without oral cleft. You also heard from Rich Platt this morning, and also Marsha Reichman, about adding Hospital Corporation of America to a Sentinel Data Partner Group. And HCA provides comprehensive inpatient and transfusion data. So it is an important data partner for our purposes in order to evaluate safety of blood and blood products. HCA covers about 5% of all hospital stays in the US, and it's important to note that most transfusions occur in hospital settings. HCA provides granular information for blood component and blood-derived products. It has EHR-based system, which is rich in clinical information as well as in patient data. And also it has ISBT and CODA-bar codes for transfusion purposes. Now, using the HCA data, we are working on another project with the outcome of transfusion-related acute lung injury or TRALI, which is the leading cause of transfusion-related fatalities. And it is defined as an acute lung injury or acute respiratory distress syndrome, which occurs either during or within the first 6 hours after blood product administration. The estimated incidence of trolley is somewhere between 1 in 1,300 to 1 in 5,000 transfusions of plasma-containing products. This is a protocol-based assessment with the objective to evaluate incidence rate of trolley after plasma, platelet, and packed red blood cell administration. The feasibility project is ongoing, and the next step is to develop the protocol. So in summary, we have built the infrastructure and tools for active safety surveillance of CBER-regulated products within Sentinel. To a large extent, we have integrated Sentinel into our regulatory process, and our safety surveillance activities at each phase of the product life cycle feed information into the activities of the next phase. What we are thinking about the future of Sentinel for CBER products is to expand Sentinel tools and capabilities to a higher capacity to cover more of CBER-regulated products. We will continue our methods development and also addition of more data elements to the common data model that are specifically appropriate for CBER-regulated products, continue addition of more data partners, and also use of electronic health records to the extent that we can. And we will further continue Sentinel integration into our regulatory process. And I would like to acknowledge a large number of people who have been working at the Sentinel Operations Center in the 3 functional groups, as well as our Sentinel investigators, data partners, and my colleagues at CBER Office of Biostatistics and Epidemiology, as well as our pre-market, 3 pre-market offices. Thank you.
Great, thank you, Azadeh. I'm going to go ahead and open up the floor for questions from the audience. And while you're thinking of that and lining up, there are some microphones here. I would like to thank you both. I mean, you guys presented very good examples of how the Sentinel tools are helping FDA address very important safety questions. Getting on the theme of how Sentinel and the tools are incorporated into the process within FDA. Marcia, you had a good outline of sort of how ARIA is being used, and so there's an initial meeting of FDA folks inside the agency thinking about how and when to use ARIA. And Azadeh, I liked your timeline about pre-approval, early post-approval, and later post-approval. I guess the question that I have is when does the planning start? So if you're thinking about using ARIA or some of the tools, Marcia, when does that first meeting of the FDA come together? Is it during the pre-approval? If you know a product is coming on the market, do you start thinking about how ARIA might be useful once the product is approved?
So I think it varies a lot depending on the product. In some cases, there's a product that's been on the market for a long time, and there will be an issue that wasn't expected that will come up. In the case of olmesartan sprue-like enteropathy, it came up through case reports and through work in fairs. And then Sentinel the question was, could it be looked at? And Sentinel could, and Sentinel was used as a supporting source of information. Sometimes there's an indication during the pre-market that there may be a problem. With mirabegron, there were changes in blood pressure. And so we knew that was likely to be approved, and we could plan beforehand to look at that in Sentinel. So I think it varies quite a bit.
And Azadeh, does it happen that way in CBER as well?
Yes, exactly. I think the key point is that it varies from product to product. For example, for vaccines in general, as I mentioned, the issue of evaluation of vaccine in pregnancy is probably something that a lot of vaccines are faced with, and that is something that comes up in the pre-approval process and is planned for the post-approval process. But then there are certain signals that may be detected at different stages of the pre-approval or post-approval and will be planned later on. The issue of rare adverse events is another issue that may come up in actually pre-market studies. And then with some of the additional capabilities of Sentinel, now we have the capability to investigate them perhaps later in the post-approval period.
Great, thank you. We'll remind folks, I know many of you might have questions. There are just a tremendous amount of applications of the Sentinel tools within FDA that are going on. So please come to the microphones with your questions. I'll continue to ask because I have time. So one of the questions, we didn't talk about this ahead of time, and it strikes me as the REMS program. When a product is coming to the market and there is at least some safety concerns that are somewhat known, not well characterized potentially. Are there any discussions or plans to incorporate some of these Sentinel capabilities in the REMS toolbox? So as you're thinking about the product as it comes on the market and the capabilities of Sentinel, can that be incorporated within a REMS program?
So we've looked at use of the REMS Etasu products within Sentinel, and for most of them the use is fairly low, but still if there were a high incidence issue, we would be able to look at it. Also, a number of the REMS products, there are recommendations or requirements for some type of monitoring, and that fits into the project that we're starting for more general situations where it's desirable to have some type of monitoring, and we want to look at the frequency of that. So that's one way of handling it.
I don't have anything extraordinary to add to what Marcia said. Again, it varies from product to product, and also it really depends on the capabilities of Sentinel for any particular issue that has come up.
So Azadeh, I liked your final slides as you were describing HCA and looking at blood products. I know the question of how to utilize the more traditional Sentinel data, i.e. the claims data, and identifying blood products has been quite challenging. Typically, health plans don't, pay for the blood product itself, so it's hard to identify codes for sort of whole blood versus red blood cells, et cetera. And so, efforts have gone into trying to find other ways to identify. So maybe there's a procedure code that might indicate that blood was given. But it appears with HCA that there's a tremendous opportunity to get a better sense of identifying the specific exposure, at least as it relates to trolley. What was the exposure? But also, I guess the other sort of complication above all of that has been also sort of lot, batch processing. And so can you talk a little bit about how the HCA data is helping with some of those issues?
One of the main, I think, advantages that HCA data brings into the safety evaluation of blood products is the specific ISBT and CODA-bar system codes that are used within the HCA electronic health records. So those would specify, I think, a lot of information about the specific blood or blood product or blood component that is being used within the inpatient setting, where most of the transfusions occur.
You have a question? Please identify yourself.
Dr. Karim from Fresenius Medical Care, North America. My question was, as we know, the adverse event reporting in the FDA is a passive surveillance system, and there is under-reporting. We really don't have the exact, patient exposure, and as such, the denominator is not very correct. So the Sentinel system, will it sort of meet that requirement, because you have all these data coming from different sources?
Yeah, that's a great question. So for those of you who didn't hear it, the question was that the traditional passive surveillance systems that FDA has been using are great, but have limitations in terms of the getting the denominator or population at risk for various exposures and safety events. So the question is, has Sentinel been able to essentially fix that problem? And I think it's a great question to both panelists.
So I think the two systems work very well together, that they supply different types of information. FAIRS requires someone to think, oh yes, there's an adverse event, and to take the time and effort to report it. In Sentinel, we look at the exposure, and then we have a record of the medical treatments and outcomes that occurred. But there'll be things that come up through FAIRS, and then we can explore them in Sentinel. There may be some things that we see in Sentinel and we want to go back and check in FAIRS. So I think it's really important to have both and have them work together.
And what I would like to add to what Marcia said is that the Sentinel database provides the denominator data for us in terms of the utilization of medical products and also in terms of the population in which we are performing our safety assessments. So with that population-based database, we are able to generate incidence rates that can be somewhat generalized to this specific population covered in the Sentinel database.
Thank you very much. I think it will be a great tool in the post-marketing arena because, it's a very heterogeneous population.
Absolutely.
Signals mainly emerge in the post-marketing phase rather than in the pre-marketing or peri-marketing, where it's very, very homogeneous, well-defined.
Right. Great. Thank you for your comments. I'd like to thank both of our panelists. We've ended the session, so thank you so much for all of the details about specific tools and how they're being implemented within FDA. Thank you. So while we're making our way we're going to jump right into the next session. So I'd like to invite the panelists for our next session to go ahead and join me up on the stage. In addition to a tremendous amount of activities that Sentinel has been involved in over the last year in using these tools for FDA safety questions, there are equally important other activities happening within Sentinel that are focused on improving data resources, developing new methods and analytic techniques. So we saw a picture that Rich had presented of the Sentinel toolbox. So how do we get those tools and what's in the Sentinel toolbox? So in the next session, we'll hear from investigators about some of these new data sources and innovative methodologies. We're joined by Jeff Brown, Associate Professor and Director of Scientific Systems in the Department of Population Medicine at Harvard Medical School and Harvard Pilgrim Healthcare Institute. Bruce Fireman, Biostatistician in the Division of Research at Kaiser Permanente in Northern California. And then Martin Kuldorf, Biostatistician in the Division of Pharmacoepidemiology and Pharmacoeconomics at Brigham and Women's Hospital at Harvard Medical School. Thank you all for joining me. Jeff, we'll turn to you to kick off, and you're welcome to either go up to the podium. Okay.
I'll go wherever you would like. We'll see if. Oh, there we go. So, thanks, Greg. This is it's an honor to be here. I've got Bruce and Martin sitting next to me. There's two of my favorite folks to collaborate with, and also I don't really like them to be together in case there's an accident. They really should be separated because it's quite a brain trust, and so we'll try to be safe. So I'm going to take a little bit of time to talk about new data sources, enhancing data capabilities. You've heard some of this before. I think I might be the fourth speaker to talk about the HCA data. I'll frame it a little bit differently about how we bring a new data partner on board and kind of how we start to use them. So to take a quick step back, Sentinel uses a set of different kinds of data sources to answer the questions. So answer the questions FDA poses. The way we tend to think of it is you have to match the question to the data, to the method, and now I'm adding to the timing, right? Do you need it in a week, a month, or a year? Kind of changes how you would design a system and things you can do. So most of the Sentinel data at this point are administrative data. It's really health plan data, think claims data. A set of folks also have clinical data, and I'm going to be talking about a new set of clinical data. And we also have used registries in the past as well, linking to birth registries or immunization registries. So those are typically one-off projects because you need to do it just for that project. So The two, and I think Jenna mentioned this morning, kind of the two areas I'm going to talk about, are the two new data sources are the CMS data and Hospital Corporation of America. So for CMS, first, FDA has a really very long history of working with the CMS data, doing some incredible work with CMS data, sophisticated analyses, and this is, I'll describe how this is separate from that work. So CMS, an insurance-based system. It's what we're bringing in is the Medicare ABC. So it's excluding the Medicare Advantage plans, which we get from the private insurers. So this is not the Medicare Advantage folks. Duke is going to be, Duke Clinical Research Institute is really going to be our proxy as the data partner. It took a long time to negotiate the arrangements to get access to the data. We're going to have access through the virtual resource data center, some people call it the enclave. The critical difference here, the critical element here is what we're trying to do is bring the CMS data online to enable FDA to use the tools that have been talked about. So that's the critical difference. We can use those routine tools with the Sentinel data by bringing CMS on board in this way. And we're hoping to have it fully on board in the fall of 2016. Because CMS essentially is an insurer like most of the other data partners in Sentinel, bringing them on board is adding an incredibly new resource and a new cohort, but it's pretty straightforward in terms of once you can figure that out, the same query tools should work because it's an insurer like the others. Hospital Corporation of America is a little different. So HCA, you've got a little background on HCA already. It's a big national hospital chain. Azadeh actually nicely mentioned these new data sources that are there. It's really inpatient medications and transfusions are the new areas that we now have access to that we didn't have before. The reason you've heard HCA so many times today is because we're actually all really excited about this new source. I mean, it's really, I'm going to have to hold us back a little bit so we can learn how to use it, but it's really pretty exciting. So there's a new kind of data in there. Probably better access to electronic medical record, so we can able to actually go look at the record a little more easily. And the difference is the unit of analysis and now the hospitalization. So within HCA, you have an admit date and a discharge date, and we're very good at looking at what happens between those dates. But that's a very different scenario than a health insurer, which has a different unit of analysis. So that's what we have to think about, and I'll describe how that makes it a little different. So how did we use the HCA data? We're transforming it to the Sentinel data model, and we added to the Sentinel data model to account for these new data sources. And I expect we're going to continue to modify the data model to keep incorporating the expanded kinds of data that HCA has. We mentioned Trolley. Azadeh mentioned the Trolley study. The reason it's such a nice example is the exposure and the outcome will happen during the hospitalization. So that's the key element, and that's perfect for this kind of data. That's this idea of matching the data to the question. This one worked out well. We have this added benefit of, because FDA's doing this Trolley study, we're learning about the HCA data, essentially cheating a little bit. We're learning about it before it had come online as a kind of easily queryable data partner. So we've learned a lot as we're doing just that Trolley study. Everyone likes numbers, so what we've been doing with HCA at this point to bring them on board as a full data partner, not kind of just doing one study, but bring them on board as a data partner we can do routine querying with. You do a whole transformation process, and then you do a lot of data checking. So I took these numbers from the data checks. We have about 4 years of data that have in the data set at this point. A lot of hospitalization, about 7.5 million hospitalizations. Inpatient meds, I double and triple checked this number. It's almost 660 million records, right? So you can imagine in a hospital, there's a lot of records for the meds. And inpatient transfusions that have been mentioned. We picked just some other things that might be of interest, kind of number of kidney transplants we see. And this is what we see in the data source that they've extracted for Sentinel. This isn't across, this is across those 4 years, and this is just based on the data checking that we've done. So kidney transplants and bone marrow, about 220,000 deliveries a year. Really nice resource for that, and we have the inpatient data for those deliveries, and 425,000 adult ICUs, adult inpatients with at least one ICU day. So where do we go from here? A week ago, this heading said initial data characterization to be completed soon, something along those lines. It's done. We're happy to report that in the last 10 days or so, the folks back at the Operations Center have gotten through that full data characterization, a lot of work from HCA, collaboration with them to do it. So it's available for querying. So now we're starting to try to use those data. So it's available for ad hoc querying now, which means we're going to figure out how to use it. We're going to learn as we go. And then the next step is figuring out the routine analytic tools of how you use it more as part of a routine querying kind of approach. The trolley project will help us do that, just learning. And then we will be doing some exploratory queries. What does it look like over here in the data and over here? You have to learn about these new sources. So that's a lot of what we'll be doing, with the expectation that we'll build tools to easily use those data and then probably keep enhancing that model. We've only really focused on a small piece of the HCA, those available data at this point. There's more things we haven't done than have when you think about an EHR in the inpatient setting. So I imagine we'll expand based on FDA's needs and interests of that data model. Thanks.
Okay, thank you, Jeff. Next, I'll turn it over to Bruce.
Okay, I'm happy to be here today to talk about plans for using simulation experiments to evaluate Sentinel's analytic tools. And these are just plans that have been getting underway recently. For some background, Sentinel's area system needs ready-for-use tools to conduct valid, reliable, rapid safety surveillance. And you've heard quite a bit about area in earlier presentations. An area includes some tools for propensity score matching designed to reduce confounding in safety studies using observational data. We're going to be doing simulation experiments to evaluate how these propensity score-based tools perform in a range of realistic scenarios. And we're hoping that our findings should clarify strengths and limitations of these tools and guide enhancements to them and development of additional tools. We have three specific aims. First, to evaluate the tools that are already in the toolbox. And as was mentioned earlier, they can do either one-to-one propensity score matching or one-to-n matching where n varies. Second, we're going to consider additional ways of using propensity scores, and these include fine stratification, full matching, weighting, as Marcia, I think, alluded to in her presentation, propensity score regression, and some doubly robust methods. And third, we want to develop infrastructure for conducting realistic simulations known as plasmode simulation experiments, because we think it's a promising way to evaluate the performance of Sentinel's methods and tools in the Sentinel data environment. So we're going to start by using data from the Commercial Claims and Encounters database at the IMED's Research Lab. And this is a secure interactive setting with Sentinel-like data that's real data, but doesn't overlap with Sentinel data. And we're going to start with an example, which would include a target drug and a comparator that we're comparing that drug to, and the new users of that drug with their, for example, rivaroxaban and warfarin, new users of those two anticoagulants with their users' baseline covariates, patterns of drug usage, and health outcomes of interest. Then we're going to sample real patients with their real covariate profiles. We're going to do sampling with replacement. And that's what plasmode simulation does. Its strength is that the relations among the covariates stay real. And then for each of many scenarios, we're going to be challenging these tools by seeing how well they work in many different scenarios. And in each scenario, we're going to artificially give patients propensities to use rivaroxaban in our example, and risks of health outcomes. And so we'll know the true risk because we'll assign it according to our own data generating mechanism. And we'll see how well the tools detect and estimate the true relative risk. So we're going to build thousands of simulated data sets for each of a range of scenarios. And we use a propensity score-based tool on each data set to estimate the target effect, typically the relative risk associated with use of one drug versus the other drug. And then we evaluate how valid and reliable were the tool's estimates in different scenarios, different ways that challenges that we've put into our scenarios. We're going to have scenarios where the drug's effect on risk is moderate or large, and also scenarios where the drug's entirely safe and there's no effect on risk. We're going to have scenarios where there's strong confounding by the measured covariates, and also scenarios where the confounding is moderate. We're going to have scenarios where the baseline risk is high, low, or very low, rare events where perhaps we can't estimate the relative risk, but we can say something about how much reassurance there can be about what the bounds on the attributable risk might be. Thus, each scenario has a made-up drug effect on risk that our tools should estimate accurately and efficiently despite the challenges we pose. We're going to assess performance with measures of bias. That's how whether on average, if we have thousands of risk estimates, whether they hit the relative risk accurately on average. Precision, how much spread there is. If there's very little spread, we can get a tight confidence interval around our estimate. Mean squared error, which takes into account both bias and precision by squaring deviations from the truth rather than from the mean of all the estimates. And the next two measures assess type 1 and type 2 error when we make hypothesis tests. What's the chance of a false positive safety signal? What's our power when we use this tool to detect a real safety problem if there is a real problem? And then finally, two criteria for good performance for a tool that are less statistical and quantitative. We want tools that are feasible in Sentinel's distributed data environment and given the privacy requirements. And we also want tools that are credible, transparent. So we're going to say something about how easy it is to understand. I want to say a little more about the challenges our scenarios are going to pose. We're going to put in there heterogeneity and drug effects across subgroups and across levels of baseline risk. Change in drug effects by time on the study drug. And we're also going to put in there what's sometimes called informative censoring, where duration of drug use associated with lapses in adherence may be associated with some potential confounding. Eventually, not at first, but we're actually going to put in some of the challenges posed by periodic analyses of accumulating data because such analyses may be needed for timely safety surveillance of a new drug. There are also challenges posed when we have more than one outcome of interest for a given target drug and when we have more than one comparator of interest. And of course, the huge problem in studies of observational data is confounding by unmeasured factors. Here's the work group members that we have on this team that's being co-led by Ling Li and myself. In red, we have people from Harvard Pilgrim, the Sentinel Center, Operations Center. We also have collaborators from Group Health, from Brigham and Women in green there, and in blue are FDA collaborators. We also have expert consultants from academics and from the data partners, and we have a timeline for this work that just began a couple of months ago and will extend to May of 2017. I have just a couple of concluding comments. I want to emphasize that the questions and issues we're addressing arose from Sentinel activities and many Sentinel activities in recent years that have been using propensity score-based methods and safety surveillance. Among the different challenges and methods that I mentioned, we're going to prioritize according to FDA's concerns and priorities. And in summary, we hope and expect that our simulation experiments are going to help FDA understand strengths and limitations of the existing tools, develop additional tools, and conduct valid, reliable, rapid safety surveillance of medical products. Thanks very much.
Great, thank you, Bruce. I'll turn it over to Martin next.
Thank you very much for inviting me here, and also I want to say it's such a great privilege to be part of the Sentinel project. It's such a cutting edge, both in terms of public health and science. I'm going to talk about a tree scan statistic and attempt to cast a wide net to detect unsuspected adverse reactions. And this is collaborative work with a bunch of people, which I will show in the last slide. Sentinel's focus has been and will always be and should always be on looking at specific hypothesis of does this exposure cause this adverse event or not. But Sentinel is such a valuable resource so that we could also use it for signal identification or signal detection. So the question then is, how can we detect any unsuspected adverse reaction that we wouldn't even think about? And also, how can we try to ensure that there are no unknown adverse reactions there that we don't know about? So one way to do that is to use what's called a tree scan statistic. So what it does is for selected vaccines in this example, but it could be drugs, we use Sentinel data to evaluate thousands of potential adverse events and groups of closely related adverse events. We evaluate multiple potential risk windows of when this adverse event may happen after the exposure, and we adjust for the multiple testing to ensure that we don't get a whole lot of false positives. So one issue when we do this is the level of granularity in the outcomes, because suppose there is a drug and there's an increased risk, that could be in a very specific diagnosis like acute liver failure, or it could be a whole range of related diagnoses of different liver problems. So in one person is one type of liver problem, and another person is other. And if we are looking, we don't know what we're looking for, we have to sort of look both at a very fine granular level and a more broader level. So what we do is we can use a tree. We have a tree with all the outcomes. So here we are, for example, at the bottom of the leaves of this tree, we have heart attack, cardiac arrhythmia, and cardiomyopathy, which is heart problems. And then we have kidney failure and kidney infection, which is another group. And then we will cut this tree, so when we cut further down, we see it is an excess risk of kidney failure. If we cut further up, is there an excess risk of any type of kidney problem? So at the lowest level on this tree, we have about 6,000 ICD-9 codes. And then we adjust for the multiple testing of looking at all these 6,000 plus groups of these 6,000. And I'm going to give one example. We have tried it on different types of outcomes, but I'm going to give you an example of the human papillomavirus vaccine, Gardasil. And we're looking for medically attended adverse events. We use what's called a conditional tree temporal scan statistic. This is a self-control method that adjusts for all time-varying confounders. And we look at the first dose of this vaccine, and we had 1.9 million doses, so 1.9 people exposed, and the data comes from 4 of the Sentinel health plans. And we don't know if this adverse event will happen soon after vaccination or further down the road. So what we do is we follow them for 8 weeks, and we say that the risk window of excess risk will start in the first 4 weeks, and it will end during the first 6 weeks. And we'll have a minimum of 2 days and maximum of 4 days risk window. And the comparison window is those days in this 1 to 56 that are not in the risk window. So this is the results for the Gardasil HPV-4 vaccine. And you can see here that if you start from the bottom, we find a signal for cellulitis and abscess of upper arm and forearm. That's one of the ICD-9 codes. That's sort of a skin issue, and that's a known adverse reaction to the vaccine, that on the injection site you have a little thing on the skin, so that's not surprising. And we would have expected to see that. So it picks up this known adverse reaction. You can also see that at the higher level of the tree, at the level 12, disease of the skin and subcutaneous tissue is also significant, but it's driven by different skin issues that might be coded slightly different by different physicians. If we go further up, we have another signal for other complications of surgical and medical procedures, including post-vaccination fever and serum reaction due to vaccinations. So that's also something that you would expect to see. And those were the two signals that we got from looking at many, many thousands of things. So what did we not find? Well, for example, these are some of the things where there was an excess relative risk, but there was not statistically significant. So these are not any signals, but it gives examples of what the method actually was looking for. So dysphagia, difficulty in swallowing, there was no reason to think that that would be caused by the vaccine. There's still no reason to think it is, but we looked for it, as well as these other things, some of which I never even heard of before doing this study. So in conclusion, the self-controlled 3 temporal scanning worked well for the HPV-4 vaccine. We found known adverse reactions. There were no false alerts. We have high power to detect rare adverse reactions, and you can see that, for example, that for the other serum reactions due to vaccinations, there were 11 observed out of 1.9 million, so that's an attributable risk of 0.6 per 100,000, so 6 per million vaccinees. That's very rare, but it still picked it up here with a pretty small p-value. So we have good power to detect rare adverse reactions when we have this huge Sentinel data set. It adjusts for the multiple testing, and that's why we didn't find a lot of false positives. We only looked for, in this case, early onset adverse reactions that happen within a few weeks of vaccinations. And we only looked, in this case, at the first dose. We used a software called TreeScan that has been developed as part of this project, and that is free for anybody to use. That can be downloaded. And in the future, we think it may be possible to integrate this into the standard 18-month product review, which Asade mentioned in her talk. It might also be used for drugs as well as vaccines, and we're currently testing the method to scan all drugs for specific adverse event with people at CEDAR. So I'd like to acknowledge the TreeScan team members who has been part of both what I showed in this talk, but also other type of TreeScan projects. I would especially like to mention Catherine Yee and Judy Mara, who is leading the HPV-4 pilot project. So thank you very much.
Thank you, Martin. I'd like to thank the other panelists as well. We're going to also have time for audience Q&A at the end of this session. So as you think of your questions, please come up to the microphones. Before we go to the audience, I do have a couple of questions for the panelists. These are all excellent examples of how tools get developed within Sentinel, what motivates their development, what the potential uses are. So my first question is related to again, it's an important topic. HCA as a data partner, so I'll bring it up again. So this is to Jeff. As I looked at the map of the United States and looked at where the HCA hospitals are, a lot, states that came up, Georgia, Virginia, Colorado, California. Those happen to also be states where there is a large proportion of health plans that are already in Sentinel, so like Anthem and United, et cetera. So my question is, how are you thinking? So presumably, the patients who are going to those hospitals, many of them probably have Blue Cross Blue Shield, United, Humana as their insurer, and so they might be the same patients. So so the question is, how are you thinking about the overlap, because you're adding not necessarily new populations to Sentinel, but rather new data pieces on the existing population. So are you thinking about that overlap, or does it not matter because of the way that you're using the HCA data?
Sure. So there is overlap, so we know that there will be the same patients in these data in the data sets. The first solution is don't ask the same question of both. And because you would ask different kinds of questions in the HCA data, which has basically a time window of the hospitalization, versus the rest of Sentinel, where the time is defined person time in the during that enrolled period. So you would use them for different questions. But you raised a second point of, well, it would actually be nice to be able to link them, because that's really what you would want to create a more robust data source. So I think we spent a lot of time thinking about how we would do it, and HCA is probably a nice. We have to. Look, I get ahead of myself sometimes, so linkage would be great. First, we got to learn how to use the data and build the partnership with HCA. But at least it's a one-to-one linkage, actually, the way if it was HCA to the insurers. There's no risk at this point. I mean, there is no risk of us finding the same outcome in the same analysis, that we would just not do that.
Can you also talk about sort of what you're learning in terms of, we've heard, and this is not new, that EHR data are great, but they're also dirty in different ways than claims data. So there must be a tremendous amount of learning in terms of how to glean the right kinds of variables that you need for an analysis from the medical record. So you can talk a little bit about that learning curve and how you're approaching that?
Well, we started pretty simple, I think, is the idea. Start with a common data model. So start with the information in HCA that's best understood by them and us, and build from there. So what we did have to build a inpatient medication table, it's different than outpatient dispensing. I mean, there's different elements in how the data are organized and the data flow, and there's an inpatient transfusion table, right? So that was important to FDA, so we built that out now. What I expect is that we'll keep learning about the HCA data as HCA data as a resource, matching that with FDA needs. So transfusion wasn't an accident we ended up at transfusions. Transfusion because it was a really important topic for FDA, and then we'll kind of move from there.
Great, thanks. So Bruce, you had plasmode simulations. Exciting. So, I understand the overall goal of that is that, there are existing techniques and methodologies that are used within Sentinel now. The simulations are one way to get a better sense of as you mentioned for FDA, the strengths and limitations of those tools. I suspect that the tools aren't one size fits all. So it's not necessarily a question of is one tool better than the other, but rather in what scenarios and what kinds of questions do some tools perform better than others. So are you hoping that the results of these analyses are going to help map out, when we see a question like this or in this kind of clinical scenario, let's use this tool, not the other? So is that sort of the longer term plan for these kinds of evaluations?
Yes, that Thanks. That's our intent. We're posing in the scenarios that we study with our simulation experiments, we're going to pose different challenges to see if the tools get the right answer, get the right estimate of the relative risk. And we expect, as you suggest, that the tools we already have perform well in a wide range of situations. But there will also be situations where we may find that it would be helpful to refine these tools in certain ways and also to build some new tools. And we're going to be focusing on propensity score-based tools, but propensity scores can be used in a variety of additional ways to the ways that have been implemented with our current tools. And we're going to see if, hopefully we'll come up at the end of this with some guidance and recommendations about situations where additional, more refined tools or additional tools would be helpful.
So you brought up the ability to use the IMEDS database, and that's useful, I think, because it's a de-identified database. You're able to get your hands on the data and run some of these simulations. Are there any plans that, once you have your answers from that data to be able to turn to the actual the actual Sentinel system data partners and sort of test those assumptions on the Sentinel data themselves?
Well, we think that for simulation, we're starting with real data, we're resampling from real data. This is a laboratory that's accessible for this purpose, and we think that the data there are Sentinel-like enough that we can make inferences from what we learn with the IMEDS data about what's going to work in Sentinel. And these questions, though, have come up in our real studies of Sentinel data, and we'll be eventually going back to Sentinel data. But given the distributed data environment and so forth, it's helpful to have a lab like that provided by IMEDS to do the simulation experiments. We have discussed, though, some tests that we could do in Sentinel data itself.
Great, thanks. So Martin, this is the, I think, as I think back to previous Sentinel meetings, the question always comes up, what about hypothesis generating? What about signal detection? If there's a new product on the market and you're not sure what to look for, then what does Sentinel do? And the answer has been, well, we don't have great methods for that yet, so we're focusing over here instead of on signal generation. But now we have, you're looking into these methods, you're evaluating these methods. You brought up a question about false positives. And so I want to ask you about sort of tolerance for false positives versus false negatives. So if we didn't have the Sentinel system, for example, we might be worried about false positives because that might alert to the public there's a concern when in reality there isn't. But we have Sentinel. So can you talk about how or at least the thinking behind when you're doing some of these signal generation or the signal identification analyses, where the tolerance level on false positives versus false negatives is, knowing that if there is a positive from the tree scan method, you can then turn to the traditional uses of Sentinel and really refine those signals.
So thank you for that question. I think in terms of false negative, what's good is that with the Sentinel, we have such large sample size that we are really able to pick up very rare adverse reactions. And if we, like in this example, we could pick up things that's 5 to 10 per million. If it's less than that, even if it's serious, it might not be so important from public health. So I think in terms of the false negatives, the sample size really helps with that. The false positives, what the tree scan does, it sort of controls the false positives in terms of the probabilistic aspect of it, so chance occurrences. So in one analysis, if there truly was nothing that was excess risk, we would have a 5% chance of having a false positive. And we can put that at a different level if you wanted to. So that's the control. The thing that's the more concerning for false positive is due to confounding. And the self-control method that I explained is good because it automatically controls for any confounding between persons that's not time varying. There can still be time varying confounders. In this example that I showed, there was no such things, but for other things, we have sometimes encountered time varying confounders. For example, we looked at the pneumococcal vaccine in adults, and we found that there was an excess of spleen surgery after vaccination. So does the vaccine, pneumococcal vaccine, causes a spleen problem that then leads to surgery? Well, not really. It really is that the pneumococcal vaccine is given as a preventive method 2 weeks before spleen surgery. So there is a causal relationship. It goes backwards in time. So actually the method should pick up those things because it can't distinguish the direction of the causality. But the clinical judgment has to be used to sort of say, well, this is not caused by the vaccine. It's the surgery that causes the vaccination.
Great. So we do have a question before. So one more quick thing. I know, a lot of tool development. Is there any way for others to have access to these tools? Are there any plans to provide this information on the website, for example?
So the software that does the tree scan analysis is available free on the web at treescan.org. So that's available for anybody who wants to try it. And that's sort of one of the products of the Sentinel project to sort of make this available, not only within Sentinel, but also outside of Sentinel.
Excellent, thank you. You have a question?
Hi, yes.
Go ahead.
I'm Doris Peter from Consumer Reports. I had a related question about the tree scan results from the Gardasil study that you did. Did you pick up syncope in your study? Because that's a reported adverse event, and I didn't see it there.
That's an excellent question, so thank you very much. We did not, and the reason is when we looked at adverse events, we looked at 1 to 56 days, and
Not
syncope
0.
happens on day 0.
Right.
They're like usually like within a few minutes after vaccination. The reason we do not include day 0 is that on the day of vaccination, that can happen all kinds of things. So for example, for little kids, if we do a vaccination, we find that there is, if we include day 0, we will find that there is eye examination. That's not caused by the vaccine, it's just that you do it at the same visit. So we try not to include day 0 on as part of the analysis. And if we want to look at day 0, we usually look at it by itself, and we sort of prepare to throw away most of the things, and only certain things will be looked at.
Did you look at day 0 for Gardasil?
We did not, no.
Okay, but you will, I'm assuming, since that's a...
We might want to do that also, yeah.
Okay.
Thank you.
Okay, question?
Yes. I was struck by the fact that you didn't have a single false positive. Doesn't that raise a few questions about sensitivity, especially for some of the terminology terms that were more frequent?
So the statistics is designed so that if there truly is no adverse events, we would only have a 5% probability of having a false positive. So 5% of the time we will have a false positive, and 95% we should not have it. So it's natural not to have it because we adjust for the multiple testing. Of course, when we adjust for the multiple testing, potentially that could lose power because you have to sort of adjust that away. On the other hand, because of the sample size is so huge in Sentinel, we still have good power. So I think it's a good thing that we didn't have any false positive. I think that shows that the method actually worked in terms of taking care of the multiple testing.
Okay, great. Last question.
Hi, my name is Khadija. I'm with Industry. Quick question. If you step back and look at from a data management perspective, this is a new phenomenon. You are taking very disparate data from different sources and bringing it together. As a side, aside from the safety application, is there going to be expectation that there'll be papers published out of this on the whole philosophical change? Because every source, data source you're using is a dynamic data source. And how are you handling that change management? Because this is starting a new trend from data revolutionizing, if I can use the word. So can we expect some papers coming out of the university on that topic?
It's for me, isn't it?
Any takers?
So we spend a lot of time worrying about the diversity of the data partners. We have this and I see it as a benefit. We have big and small, they're all over the country, they have different systems, different policies even, which we can benefit from. We spend an awful lot of time going through essentially a standardization process and a checking process. I won't bore you with the data checking cycle, but every time a partner updates their data, we go through, it can be up to 1,500 checks to ensure some consistency. But that only gets you so far, right? I mean, it's really important to check every time. So what we do is every time we ask a specific question, most of what we're doing is checking consistency across the data partners, looking for anomalies, because you can't check every dark corner of the database. So we do this initial check and validation of the data sources for consistency to get them to the point where we think we can use them and we understand them. And questions come up and we ask and answer, and they get asked and answered. But then every time we want to ask a question, for example, if it's HPV-4, we'll go and look at HPV-4 across the data sets by age, by year, by sex. Age, year, and sex. to see if it's consistent, and if it's not, to ask questions. One of the benefits of working in this environment is that we can call the data partners, and we do, and they know it, and we have a direct relationship with all of the data partners. So when we see something, we have a phone number. We call and say, hey, in 2004, something looked funny, and we talk about it. And we can do that as part of the data checking process, or we do it as things pop up in particular analyses. If something looks unusual, we'll make a phone call, and our partners are great, and they call back, and we talk to them about it. It's different than buying the data. It's really different. These are partners that work with us on it.
Great, thanks, Jeff. That concludes our session. I'd like to thank all our panelists for joining me up here. As we said before, lunch is on your own. There are some great restaurants in the area. If you have any questions, Joanna at our registration desk can point you in the right direction. We'll be reconvening back here exactly at 1:15. The next session will start sharply at 1:15, and feel free to bring back food into this room if you don't have time to finish it outside. Thank you, and have a great lunch.